Hi, this is Miss Litton, and this is 7th period support for Chapter 8, Cellular Respiration. Say hi, everybody. Hi. Okay, so quick rundown. When you, for both photosynthesis and cellular respiration, I'm thinking like already out to the exam, you would want to be able to write the equation from left to right if I asked you photosynthesis, or write it from left to right if I asked you cellular respiration. I would say what people forget in photosynthesis, you know what people forget? Light, yeah, the sun. That's a typical thing that people forget. And I would also ask you to identify what's getting reduced and what's getting oxidized. And what I end up people screwing up is they draw it like how we drew it in class. Remember how we did it both ways, cellular respiration and photosynthesis way? Do you remember that? And they just draw everything, which tells me you know nothing, right? So if I ask you only for photosynthesis, only draw photosynthesis left to right. If I ask you for cellular respiration, only write cellular respiration left to right, and only identify what gets reduced and what gets oxidized for that particular reaction, right? Does that make sense to you? Or am I confusing you, or you got it? Got it? Okay. So we did that here, okay? So this is showing what gets oxidized and what gets reduced just for cellular respiration. All right? Another thing, we had new players. Instead of NADH as electron carriers, what did we have? NAD, and who was our other carrier? FAD, okay? Then the other thing I would get really comfortable with are all the steps, whether you're doing aerobic or whether you're doing anaerobic. Yeah, and maybe how they overlap, and I would be comfortable with that. I don't know if I've done a hideous drawing or not. No, I have not. Okay, but if I was going to do a hideous drawing, I would probably start with glucose up here, and I would, if you were home, I would see if you could do this at home, and I would go glucose to what? What does it get changed into? Pyruvate. And I would know, I would probably put down in here 2 C3s, and I would probably put up here what? C6. Exactly. What else happens? And I would use different colors, but that's what I would do. But what else happens? Reduce. Yeah, 2 NAD, sorry, reduced. I didn't do that very well. And what else? 2 ATP. Now, I know if I did that I actually had to invest 2 ATP and I got 4 ATP out, right? And then I would have to make a decision. From pyruvate, what would I have to decide? Yes, if I had no O2, I would do what process? Fermentation. And if I did fermentation, how many choices would I have? 2. I would either form lactic acid, makes our muscles sore. I would form what? Alcohol. Plus, yeah, 2 CO2s, right? Don't forget about that. And then what would happen? What would I be trying to do here? Yeah, I would be oxidizing my NAD, 2 of them. And that is this, and I know I'm doing a terrible job drawing, I'm just trying to do really fast. That would be the sole reason why you did fermentation. Sole reason is to oxidize your NAD. And the reason is, in order to walk away with 2 ATP, you have to reduce 2 NAD first in glycolysis. How many steps is glycolysis? 10 steps. So I would be indicating that at some point, you know, that glycolysis. What else do you know about glycolysis? It occurs in the cytoplasm. Good. And there are 10 steps here. Okay, there's energy investment stages, there's energy harvest stages. Okay, turn toward your bio buddy. Go. Are you good with that part? Questions, concerns? All right. Come back back to me. Is there anything you want to ask me before I go forward? I'll show you the notes and stuff. I'm just seeing if we can distill it down to the most important things. Okay? Any questions you want to ask me about that? God, it's so hideous. Yes. Yes. Is this the picture you want to have, though? Okay, go ahead. You never have to ask me. I don't have to take pictures. Okay. Now, what would be another scenario? That's scenario 1, the no oxygen scenario. Give me another scenario. There is oxygen. So, with oxygen, what's it called?
Aerobic respiration.
Aerobic respiration. Okay? And with aerobic respiration, I would take my pyruvate and I would convert it to what? To acetyl-CoA. Okay? Along the way, what would I do? I would reduce some NAD. And what else? Am I making ATP here? No, I am not. Okay? Remember, pyruvate was 2 3-carbons, or was the 3-carbon, and you take a CO2 off of it, and in its place where the CO2 was, you put an acetyl-CoA group, and the acetyl-CoA is going to help you jump into the next stage. You need the acetyl-CoA, the coenzyme, to help you get into the next step. Okay? Since I have 2 pyruvates, I'll have 2 acetyl-CoAs, I'll have 2 carbon dioxide generated. The electrons from that bond between those 2 carbons that I just separated, those will go to NAD, forming reduced NAD. Yeah. All right. Now I have 2 acetyl-CoA. Now I can go in and do the what cycle? Yeah, the citric acid cycle. I also called what else? TCA, also called what else? Krebs cycle. How many times am I going to do it? 2 times. And what is already preloaded into this cycle? Yeah, a 4-carbon, okay? Sorry. A C4. And this is a C2. What is 2 plus 4? C6, okay? Because 2 plus 4 is 6. Then we do what? We do the bit. When we do the bit, what are we doing? Did you say reducing NAD, CO2? Yeah? And then we have a C what? It's a C5 because we took a carbon away right here. Yes? Okay. Then we what? Do the bid again. And then we're down to a what? C4. Okay, then what? Make an ATP. Now all I'm showing you is the ATP, but you know I would need an ADP plus an inorganic phosphate, yes? Okay. What else happens? Reduce FAD and reduce NAD. Then I do the whole thing all over again, yes? Yep. Okay. Then what I need to do is I need to go and collect all my what? Oops, yeah, my NADs here, from transition prep, from here, and I do this twice, so I would have 2 of each of these, yes? And I take them all to the top of the ETC. Let's see if I can do this. I can do this. Okay. And yes, I would like to continue. Okay, I take them to the top of the ETC. Now, they don't all actually get to go to the top, right? No, some come in at the very top. Your NAD reduced comes in at the top, but your FAD comes in a little lower. Now, the NADs from glycolysis could come in at the top, but oftentimes they come in lower because your electron transport chain is in the mitochondria, and it might hand off its electrons to FAD and have FAD take them on in. Okay? And so when you drop off those electrons, there are then 3 spots, reductase, tell me, reductase, cytochrome oxidase, which are then pumping what? Hydrogen ions out. Yeah, so that it can come back in through, kind of looks like that. Let me just change it briefly. Sorry. I was trying to make it look like how it did in the book. What is it called? ATP synthase complex. Hashtag awkward. Okay, so then the hydrogen ions want to come back in and you make some ATP. Since FAD comes in a little bit lower, it only catches the last two, where if you come in from the top, you catch all three. Hey, check with your bio buddy. See how they're doing on that. Okay. Riddle me this. Where does glycolysis take place? Cytoplasm. Good. Where does transition prep take place? Where? Mitochondria. Matrix of the mitochondria. Where does the Krebs cycle take place? In the matrix. Where does the electron transport chain take place? The inner membrane, the cristae. Where are the hydrogen ions concentrated? Inner membrane space, the outermost space of the mitochondria. Okay. What about anaerobic respiration? What if I'm a bacteria? Yeah, where does fermentation take place? In the cytoplasm. They have membranes too, just not any membrane enclosed organelle, so they can have a concentration gradient. They can do aerobic respiration as well. Just no membrane enclosed organelles, which makes it a lot easier if you have mitochondria to organize your reactions, right? But what do you give up if you're a eukaryotic cell? Surface area, right? Right? A larger cell has less surface area per volume. Just reminding you of that. All right, so let's take a look when we have reviewed it. Okay, here is glycolysis. You walk away to ATP, go into acetyl-CoA, lead it into citric acid, do your electron transport chain. There are so many diagrams. If you just Google cellular respiration, you should be able to pull up any diagram and go, I understand it. Okay, you should not have a problem with that. We're good? I'm just reminding you, I'm showing you a few things. Remember, it's really 10 steps, right? And then, let's talk about the different kinds of phosphorylations that we know. What's the first phosphorylation I taught you? Photophosphorylation. In the what? Light reaction of the chloroplast, right? Okay, on the what membrane? Is that the first place I taught you that? Yes, that is. Photophosphorylation. I was like, thinking for a minute. Photophosphorylation. Okay, give me the next one. Substrate level phosphorylation. Where do we do that? We do that in glycolysis. Good job. Where else do we do that? Krebs cycle. Good job. Remember when we make NATP in the Krebs cycle? That's substrate level phosphorylation. What's the third one? Oxidative phosphorylation. Where do we do oxidative phosphorylation? Where? At the bottom of the ETC, which is a critical part that we left out of our diagram. Where did I put that diagram? Here it is. Yeah, at the bottom of the electron transport chain, which I screwed up with my ATP synthase complex. Oh, look, there it is. Okay. At the bottom of the electron transport chain, who catches those electrons at the very bottom? Oxygen forming water. Why do we care about forming water? Do we care about forming water? No, we just need a way to remove the electrons, right? To remove the electrons that went through the electron transport chain, because they can only hold, each little step on that electron transport chain can only hold so many electrons. So if you don't have a way to let them out, right, then the whole thing gets backed up. So how do we take care of that when we do photosynthesis? Because we have 2 electron transport chains there. How come it doesn't get backed up? Ultimately to reduce the NADP, right? But When we look at it from left to right, photosystem 2 donates electrons to photosystem 1, and photosystem 1 donates its electrons to NADP to form reduced NADP. Okay. Good. I think we're good. You comfortable with this? 2 strategies to oxidize your NAD. Does it have to stay permanently as ethanol and lactate? What could happen? What do? Yeah, we convert the lactate back to pyruvate. And we reduce NAD in the process. And then send it through when we have oxygen. Our muscles do not stay permanently sore, right? We remove that from our muscle tissues. Okay, we know about the structure of the mitochondria. We feeling good about this? Feeling good about this? Okay, so gather all your reduced NADs and FADs and take them to the top of the ETC, and then it's and then what? Okay, then we say na 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 na, and this is what? Reductase, cytochrome oxidase, and you make some ATP, a lot of it, or what? A lot of it. If you drop off the FAD, who catches it at the bottom? Oxygen forming water. Okay, do you understand that diagram okay? You good? And I showed you this one. We looked at this and we did the math. Remember that? And this was wrong right here. Are we good with our math? Need me to review that at all? Nobody saying yes? Review? Okay, you got it. So, cash money. I think of ATP as cash money, and cash money comes from substrate level phosphorylation. I get 2 ATP. I walk away, right, with 2 ATP. I walk away with 2 ATP when I do the citric acid cycle 2 times, right? So that's cash money. So 2 there, I've got 4. Right? From glycolysis, though, I get 2 reduced NAD. Now, if they go in at the top of the chain, they could get me 3 ATP because they would get the reductase, cytochrome oxidase, yes? But if they go in the bottom of the chain, partway through, they're only going to get reductase, cytochrome oxidase, yes? So they'll only get me 2. So it's 3 or 2, right? That's why we say 2 times 2 is 4, 2 times 3 is 6. Somewhere between 4 to 6 ATP when you cash in the NAD at the bank of the ETC. Yes? Okay? Now, transition prep, right? Reduce some NAD. Those 2 NAD here, those are going to come in strong at the top. So they're going to get us 3 ATP each. 2 times 3 is 6. Okay? In the citric acid cycle, do the bit, reduce an NAD, that's 3. I do it 2 times, so that gives me 6 total. That's where this 6 is coming from having done it 2 times. 6 times, they're all strong, 3 is 18. FAD comes in lower, so each one I reduce an FAD the next time I have 2 of them. They come in lower. 2 times 2 is 4. So when I add that up, it's 32 or 34 plus 4 gives me 36 to 38 ATP. But that is if I ignore the second law of thermodynamics, because we know FAD only gets you about 1.5 on average, and an NAD only gets you 2.5 on average. All right, questions you want to ask me about that? Okay, and then I showed you pictures like this. That's a great picture. It puts it all together and way better than the one I drew. And then there's this one, also way better than the one I drew. Okay, which would be good. We also talked about the process of catabolism and anabolism. Catabolism is when you break things and release energy. You can couple that with an anabolic reaction that requires energy, right? And did I ever push start? Oh, yeah, I did. Okay. We talked about that molecules like glucose. We learned it, doing it with glucose coming in at the top with glycolysis, transition, Krebs cycle, electron transport chain. But everything we eat can come in at some point in this cycle. Proteins can come in as pyruvate, acetyl-CoA, and citric acid cycle. Fats could come in, the glycerol could come in a little bit as a precursor to pyruvate. Those long fatty acid chains, 16 to 18 carbons long. If you had 16 carbons in your fatty acid chain, how many acetyl-CoAs could you get out of that? 8, and if you have 3 chains, then that would be 24 acetyl-CoAs. That's why fat has more calories than carbohydrates per unit, right? Per one glucose molecule, per one fat molecule. Okay, and then we compared and contrast photosynthesis with cellular respiration. We talked about a really good mediator for cellular respiration or regulatory fructose 6-biphosphate. That is what this is an intermediate step, but you need phosphofructokinase in order to make it. And if you have enough ATP, so you don't need to do cellular respiration, you have all the energy you need, then the ATP will actually interfere as an allosteric inhibitor binding to a secondary site and make it so phosphofructokinase is no longer functional. And that way you stop it early on in glycolysis. You're not waiting later on in that reaction. So that was an example of what kind of feedback? Negative feedback. Or what's another one? I taught you another one. Type of inhibition? End product inhibition. Yeah, your end product is ATP. So let that end product inhibit your enzyme. We then talked about cellular fitness, okay? And there's nothing in your textbook for this. I mean, it's not in any one place, so if you're looking for it, you're not going to find it. It's just in the notes that I'm giving you, and that's because it's kind of a new thing that they're kind of adding in this year. And okay, so to specifically point it out. So cellular, we think of fitness like how fast you can run or whatever as fitness, but we know in biology terms, fitness is your ability to pass on your genes. So your cells being fit help you to pass on your genes. Do you agree with that? So it starts at the cellular level. So by having a wide variety of molecules, you can better respond to your environment and survive in different conditions by having a wide variety of molecules. And therefore, if you can respond to your environment better, you're more likely to survive and reproduce, right? So we looked at different molecules that we have, the variety within them. We, of course, know about variety in DNA. That's the obvious one. Genes and variety in our DNA gives us different traits and different adaptations. Taller, stronger, faster, rounder, whatever it is that you need to be for that particular organisms. The different polysaccharides, if you think about you could use polysaccharides to build a cell wall, or you could build polysaccharides to build chitin, or what? Give me some more. Starch. What else? Cellulose. Good. All of those things, right? Glycogen. Those are all different varieties of it. We looked at the MHC markers, right? Major histocompatibility markers, the glycoproteins. Remember we talked about that? It's like your cellular what? Fingerprint, right? And recognition. And I told you phospholipids, that there are different types of phospholipids, and it has to do with how you respond to temperature and protecting your cells and the integrity of your cells. Another example we looked at was hemoglobin, and fetal hemoglobin has a greater affinity for oxygen than your hemoglobin here that you have now. At a certain percentage of oxygen gas, a partial pressure gas of oxygen, you would unload it, but the fetus is still loading. And that's critical for the fetus to get any oxygen from you if you're pregnant, because you need to be unloading and it still needs to be piling it up so it has oxygen to carry to its tissues. Once that baby is born, you don't want it to have the same stickiness for oxygen because it just won't work with a large organism and its metabolism, so we want to unload that oxygen at a lower partial pressure. And that's what it's showing you right here. If you come across here at 50% saturated, for a fetus this would be the amount of oxygen it would still be loading, but to have 50% saturation, you would need far more oxygen for the mother to still be loading at that level. Okay? And that's so that the fetus can get at the oxygen. And last but not least, we talked about, oh, that's just another one of those. We talked about chlorophyll. And the variety in chlorophyll and the pigments that a plant has allows it to capture light at all different wavelengths and all different seasons, which makes it more adaptive, more sugars, more likely to survive and reproduce. Okay, time to sing. Are you ready? I'll leave this one up. Okay, you ready? Both hands. So we're going to start with cellular respiration. First step, glycolysis. By the way, this is on my YouTube channel if you need it. Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do we start out with? Glucose. How many carbons? 6. What are we going to do? Break it in half. How many steps? 10. Enzymes? 10. What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. Ready? 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all your reduced NADs and FADs and take them to the top of the ETC. Reductase, reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catches at the bottom? Oxygen forming water. Water, 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 water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweet, sweet. Here comes the sun traveling in waves. Particles of light are called photons. And it gets the electrons excited. PQ. And you make some ATP. Here comes the sun traveling in waves. Particles of light are called photons. And it gets the electrons excited. Who catches it? NADP forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-independent reaction. Ready? 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take 1 away, PGAL, G, 3P. And you're left with 5C3. Use some ATP to build 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. You like the choir.
Take 1 away, glucose.
And you're left with 5C3. Use some ATP to build 3C5. Stop. Pick up your glucose. Break it in half. How many steps?
10.
How many enzymes?
10.
What are these called?
2 pyruvic acids.
And you?
Reduce some NAD and make some ATP. 2. Oh no, we don't have any oxygen. What are we going to do? Fermentation.
How many choices do we have?
2.
You and I.
Lactic acid makes our muscles sore.
Plants and yeast make
alcohol. Either way, you oxidize our NAD.
I'm so proud of you. All right. Pause. Questions. Questions. What do you want to ask me? Questions. Some of you are like coming up on this and I just want you to know. Ask your question now. What's the question? Stranger danger. What's your questions? Any Yes. Anyone? Okay, yes. Wait, louder. The children can't hear.
When you get the water from the first part of the cycle,
Which one?
like the cellular respiration, you end with water. And
Yes.
So in the second part, if you're already making water, why does it need to?
Oh, okay. So, when I link the two songs together, it's just so you can link the two metabolic songs. Now, a plant will link it together. And I will tell you, the kangaroo rat, who is like a desert organism, they get all the water they need from the metabolism of the grains that they eat. They do not have to drink their whole entire life. They harvest the water from water. Yeah, we're just not that efficient. What else? Yes?
Can you re-explain substrate level phosphorylation?
I absolutely can re-explain that. So substrate level phosphorylation is basically just passing a phosphate from one molecule to another. It's going to happen here. From one molecule to another. So it's not any kind of electron transport chain. It's like I have the phosphate, now I hand it to you, you have the phosphate. That is substrate level phosphorylation. So that's not going to just happen, right? If I have $100, I'm not going to go, now you have $100, okay? I'm not going to be, so some enzyme is catalyzing that reaction so that handoff takes place. Okay, yes?
Could you re-explain the reason why the baby's hemoglobin?
Yes, I can. Okay, so let's get a fresh diagram. There's this one, okay? Or I can use this one if you want, okay? So don't look at this. Sometimes where people get confused is they look at the bottom. We're so used to time being our x-axis that that's what you think you're looking at. Don't think about time at all, okay? This area right here where you have 100% oxygen, which you would only have 100% oxygen if you were breathing oxygen like this, right? That partial pressure. This would be like in your lungs, okay? So in your lungs, in this color blue, if you were a mama, no. If you were a mama, you would be 100%, right? Saturated. All your red blood cells would become saturated with that oxygen. This out here would be somewhere where you're traveling away from your lungs and you would have to go to your heart, but then back out to your tissues. You're starting to let go of the oxygen as you get out into your tissues because your tissues need that oxygen. So what our hemoglobin is adapted to do is when the partial pressure of oxygen is low, when the temperatures are high, when the pH is low, we release oxygen. Those are all different ways to facilitate hemoglobin letting go of it. Because think about it. Hemoglobin is hanging on to oxygen and then it changes its mind and lets go of it. Why does it let go of it? Because there's not a lot of oxygen around and things are going to go from a higher concentration to a... That's just entropy, right? That's just entropy. So we let go of it as we get out in our tissues. Okay? If you were pregnant, no. If you were pregnant, you would start to be letting go of oxygen right here. Like when the partial pressure of gas, whatever that is, let's say that's like 40. Is that okay? At 40, you'd be letting go. But look, the fetus is still loading and saying, no, I can carry more oxygen. It's still taking it. And that's good. Because if you're my baby, if I am letting go of the oxygen, she can then still take it. And now she can take it to her tissues. Does that make sense? If we both had the same level, I'd be letting go at the same time my baby was letting go, she would never have any oxygen. So that's an example of where hemoglobin, there's a variety in hemoglobin based on development. So when you're an embryo, it's one way, and when you're an adult, it's a different way. Okay, what else? Oxidative phosphorylation is what we're doing in this chapter right here. Okay? And in order to make our ATP, okay, right, what we're generating ATP, right, a lot of it, or a lot of it, and we're using a hydrogen ion gradient that was done because oxygen was the final electron acceptor in an electron transport chain. So this is telling me that I need oxygen to remove the electrons so that I can make ATP. In photophosphorylation, I used light to excite the electrons so I could phosphorylate.
So photophosphorylation leads to oxidative?
No, let me try another way. In photosynthesis, right, you phosphorylate in photosynthesis, yes? You make some ATP, right? Remember PQ? How did when you say photophosphorylation, it's saying light is a part of the process. You have to have light. If you don't have light, you can't excite water's electrons. If you can't excite water's electrons, you can't na-na-na-na. You can never make a hydrogen ion gradient. You can never make any ATP. So light is a component of that phosphorylation, so you call it photophosphorylation. In us, in our mitochondria, and in plants' mitochondria, it's oxidative phosphorylation, which means oxygen is required in order for you to phosphorylate. Our electrons come in hot because they come in as NAD and FAD reduced, right? So they come in hot. So it's not talking so much about, where in photophosphorylation, it's talking about what's happening at the beginning, and oxidative phosphorylation is talking about what's happening at the end. Does that help?
Yeah.
So you need oxygen to take the electrons away. And in substrate level phosphorylation, I'm not using oxygen or light. I'm just saying, here's my phosphate, now here's your phosphate, and an enzyme would have to catalyze that process.
It separates the electrons from the ATP?
Yes.
Oh, right.
Okay? What else? Anything? Highly suggested reading and thinking. Your quiz isn't tomorrow, right? Your quiz is Thursday, right? Isn't it? Is that when it's on the schedule? I am at a conference on Wednesday, so that's another reason why I did the review today. So I wouldn't be here the day before. And I figured you'd want to review them. Yeah? I don't know. Okay. Lab notebooks, right? We're finishing those up tomorrow. Ready to turn in? Oh, it's not a lab notebook. It's digital. Oh, yeah. I know. I can't believe I gave you that gift. Unbelievable. Okay.
So is it due tomorrow then?
Yes, due tomorrow. At the end of the class. That's all you do. What? I don't know. Okay, have a good evening.